So I will start with two apologies and an explanation. The first apology is that I'm sorry that I'm unable to give the talk in Canada. The second apology is that whenever you write a talk, you write it with an audience in mind. This talk, as it stands, was written primarily for students of biology and scientists. But it has a component that's broader that I hope that you can appreciate. So if there are sections of the talk that you don't understand, I hope that you can just hum through them like a song whose words you forget, and then you can pick up on the other pieces. The explanation is that if it looks like I have some horrible disease, it's just that we got back from the Western Ghats and the leeches were very bad. So, even though this talk was written for a scientific audience, it is not a typical scientific talk. It's about not just results, but why we do things. And it's, to a certain extent, it's a philosophical and thoughtful investigation into what we do. So I'm going to start in a personal way, not a typical way to start a talk. And the first section is Divide Creek, and it's about chance. So this is me right there, in 1970. That's my brother. This is my father. This is my cousin. I remember this day very well. I do not remember this photograph, but I remember where we went after this photograph. But we didn't take a photograph at that other place. But I managed to get there a few years ago, and so I took a photograph. And this is that other place that I remembered so well. It's called Divide Creek. And you might wonder, why would I remember a creek for so many years? Well, what I remember is the peculiar arrangement. And at the point where I'm standing, the creek divides in two. So the creek flows this way, it hits these rocks, and half of it goes right to the Arctic Ocean, it goes east, and half of it goes left, west to the Pacific Ocean. And I remember as a 12-year-old seeing this creek, being amazed with the idea that two little pieces of water that are only a millimeter apart could hit just on either side of a rock, and one of the pieces would go to the Arctic Ocean, the other to the Pacific Ocean. And what a big difference in outcomes for such a small difference in origin. How fate, how chance is so important. I should have known at that point as a 12-year-old that I was going to be an academic, that this is such a memorable thing for me, but it was quite memorable. And I think each one of us can remember Divide Creek moments in our own lives. A moment where something small happened, we made a choice, and we didn't know it at the time, but things were very different because of that small thing that happened. And for me, a Divide Creek moment happened the next year when I was playing by the shores of Lake Ontario in Canada, in eastern Canada, and a mat of grass floated by, and on this mat of grass, so presumably somebody had mown their lawn and thrown the grass onto the lake, and on the mat was a spider that looked like this. And she looked up at me, and I looked down at her, and I was startled. And I was startled because she looked up at me. I'm not used to a little thing like that looking up at me, paying attention to my world. I was always interested in nature, but this was so peculiar that I had to look at her. So I saved her from this mat on the lake, and I kept her as a pet, and I watched her behavior, and I was fascinated. And I think the reason I was so fascinated was because she reacted to the world very much like the way I thought I reacted to the world. I would see something happen, and I would look at it. She would see something, and she would look at it. Most spiders, they smell things and touch things, but they don't see very well. So they run around, and you can't understand what they're thinking. But I felt that maybe I could understand what she was thinking. So I kept her as a pet, and I became fascinated with jumping spiders. So here's a little species of jumping spider from Brazil, and he's looking around. You can see his big eyes up front. This is a female, and oh, he sees the female. And oh my gosh, what am I going to do? There's the female. Oh, maybe I'll ask her out. Okay, so here we go. He's doing a courtship display, a dance right there, but she ran away. So now, oh, where is she? Oh, look, she's over there. So as we watch him, we know that he's reacting visually. We can see the fact that he's reacting using his eyes to what's around him. So jumping spider eyes are remarkable. They have 8 eyes, the way most spiders do. Most spiders have 8 eyes, if you look closely enough. 6 of the eyes of jumping spiders are a little bit like our eyes, in that they're just round, and they see a broad field of view. But 2 of the eyes up front are quite different. They see a narrow field of view, but they see very well. So if you were to look at the back of that eye, you would see the part that receives the light is not just a big circle that receives the light and has an image form on it, but it's a strip. It's a vertical strip. And in fact, this is the retina of one of these eyes. Each of these spots is a little photoreceptor cell, and you can see that here they're very packed together so that they can resolve very fine details. In fact, these can see in terms of the fineness of details as well as a cat. And that's a remarkable thing for a spider that's 5 millimeters long. People are interested in this visual system for robotics, how to make a nanotech thing see well at a very small size. So, I forgot to explain it. The fact that this is just a vertical strip and that these are long means that it's sort of like a telephoto lens with just a small sensor. It's only seeing a tiny piece of the world. So in order to see more of the world, it actually moves the retina. It's a scanner. It scans back and forth as the spider is sitting still. So I'm going to show you this video in which you can see those eyes moving. The eyes scanning the environment. So right there, you can see how the eyes inside the head are moving around to scan the environment. So jumping spiders use these eyes to hunt like cats. They can spot a fly from a distance, they creep up, and then they jump. And in fact, the name comes from the jumping, not from the eyes, but it's the eyes that enable them to have this lifestyle of hunting like cats. They don't spin a web to catch food. So they jump. But how do they do that? Do you notice they didn't have big back legs? When we see a grasshopper, it has big back legs because, of course, it's got the muscles to power the jump in those legs. A frog has big muscles in its back legs to power its jump. How does the jumping spider do it if it doesn't have big back legs? Where are the muscles that power those legs, power the legs? So, a little closer. Okay, thank you. So how do they do it? Well, there's a hint here. I took this photograph thinking I was going to take another photograph like this, but he jumped just as I was snapping the photo. And you can see a hint as to how they managed to jump by looking at these legs. You'll notice that all these spines are standing straight up in this one, but not in this one. And so what he does is that he raises his blood pressure very quickly. And so the legs, which are curled underneath, when the blood pressure goes up, the leg snaps like this, and out he jumps. So in fact, the muscles to power the jump are in the head, not in the legs. Because the muscles in the head clamp the head shut down, the blood squirts into the legs, and the spider jumps. So I was fascinated with these creatures as a teenager, and soon I found many other species near my home. I think I can hold it. So these species I found near my home. years, I started to go further and further to look for spiders in other places. So first, other places in Canada, but eventually I went to Mexico, and I traveled quite many times to Mexico. Ecuador in South America, many times. Singapore and Malaysia, twice each. Papua New Guinea, once. Gabon, once. This is one photograph of many that I could show you of transportation incidents while doing field work in tropical countries. It happens all the time. But not always tropical countries, sometimes to temperate areas, like this one last year to Siberia. And along the way I've seen many different jumping spiders. And all of these on the screen now are jumping spiders. You can tell by the nature of the eyes. They all have the 4 eyes up front and the 4 eyes on the top, just like what I showed you. Some of them, like these, are very narrow. Some of them are very wide, depending upon the species. Some of them have lots of patterns. Some of them are very colorful. And I'll note that one of the most colorful in the world, this one here, lives in Mysore, right? This right here. In fact, there's probably one of them right now within 100 meters of here. Just like that. Some of them look like ants. These aren't all spiders. They're all jumping spiders. Some of them look ferocious. They're not really very ferocious. Some of them are cute, if you are so inclined. And along the way, I've seen many new species new to science. And so I became a spider taxonomist, describing new species, making them known. And a systematist working out their relationships, trying to figure out where they fit on the evolutionary tree. I also became a theoretician, developing phylogenetic methods, quantitative methods to help us understand evolution and the evolutionary tree. And a programmer building tools for biologists to do analyses. Even though I'm best known, as I think Kieran said, for my work on the theory and the computer programs, I consider the basic work on spider taxonomy and systematics to be my most important work. And one of the reasons is it will last centuries. If there are still people, and if there are still spiders, and if there's still people studying spiders in a few centuries, those are potentially doubtful, then people will still be citing that work. But I think my theoretical and computational work will be forgotten in a few decades. This work of discovering, the basic work of discovering biodiversity is of long-term critical importance. Those of us who discover and describe biodiversity are cartographers of sorts. We map the tree of life. We find its leaves, the species that are alive today, and we understand its branches. The work to map the tree of life does not fit the standard narrative of science. And let me explain that. We focus on particulars. We focus on particular species, particular histories. We are not looking directly for general laws. And by general laws, I mean things like E equals MC squared, or similar things in biology. And this is true. We're not looking for general laws, whether we are operating in the mode of the 19th century, drawing their body parts, or whether we're operating in the mode of the 21st century, doing phylogenomics. Either way, we're not directly looking for general laws. The standard narrative of science would tell you this is what science is. It says we go out and we gather a series of observations of similar kind. So we do manipulations on the little rats in the laboratory. We treat them in different ways. We look at their responses. And then we try to see, is there a relationship between treatment and response? And once we've done this, we derive that relationship as some sort of curve or general explanation. And at that point, the only thing we care about is the curve. We're ready to throw away those points. They back up the curve, but we don't care about the points, really. We only care about this line. We don't care that the rat had a bad day there. We don't care about any of the details. We only care about the curve. That is the standard narrative of science. The only thing that matters are these general processes, patterns, rules, relationships, laws. And yet, this was a celebrated discovery. This was a celebrated result, the human genome. And this, Copernicus's map of the cosmos nearby us, is probably considered the most important scientific results of all time. And the reason is, he put us in our place. Copernicus said, we are not at the center of the universe. We're not the focus. We're merely one of the participants and observing it from within. And in fact, Darwin did the same thing with the Tree of Life. He said, we are a participant, we are not the focus. These maps, each one is a map, these are the foundations of their respective fields. And yet none of them is a statement of general law. None of them is about how things work in general. They're a statement about particular entities and their relationships. And of this map, the Tree of Life, so much remains to be discovered. So many leaves that we haven't found yet, and so many branches that we haven't connected yet. Okay, I'm going to continue with the idea of chance, but thinking now about evolution and how it happens. So here's the Tree of Life. You are here. You're somewhere in there. With primates, with mammals, with vertebrates, jumping spiders, and over here. We can, in our mind's eyes, look back through the ancestry of both. Remember, these lines are the ancestral lineages that diversified and gave rise to all the species that are around today. So we can look back along our lineage, ancestrally, and the jumping spider lineage, and we can find a point where it's an ancestor in common to both. Now that would have been a species, not just a single individual. What would it have looked like? We don't exactly know. It would have been an animal. It would have been bilaterally symmetrical. It wouldn't have looked like a jellyfish. It would have had a head. It would have lived in the sea. So we don't know exactly, but let's just imagine it's a worm of some sort, and I'll draw it like that. So there's the little worm. It was a species of worms like that that was this ancestor in common to both. And so let's imagine, in our mind's eye, this ancestral population, generation by generation. So here's one generation. Now, of course, it probably would have had more individuals than just a handful here, but in kind it's the same. The blue lines represent descent. So this and this, mother and father gave rise to 3 babies, and this is their genes being passed down to the babies. Likewise here, and so forth. So generation by generation, this population is continuing. But at some point, something must have split it in two. We don't know what it would have been. We don't know whether it was a current that blew some individuals to another island. We don't know whether some sediments came down that divided a rocky habitat. We don't know. It likely did not happen as fast as shown here, but something divided this population in two. Now, if that's the case, which it must have been somehow, there will be a point on this diagram here where there would have been a mother worm whose descendants, whose children, ended up going on different sides of this divide, their genetic descent went on different sides of this divide. So I think you can see this is another divide creek moment. This is a moment at which 2 brother or 2 sister worms coming from the same mother ended up going in such different places, looking into the future. And what I mean by that, going to such different places, is that if we take this split, where the population was split in 2, and we fast forward hundreds of millions of years, of course the one side diversified into thousands of species, including us, and the other side diversified into millions of species, including the jumping spider that looked up at me. Now, at this point right here, this subpopulation that's recently split from this one, they're pretty much the same worm, right? They're just the same worm. And yet, they went to such different places. And so the question is evolution predictable? And in fact, Stephen Jay Gould, the well-known paleontologist who wrote natural history articles and thoughtful articles about how evolution worked, had a little thought experiment. And he was concerned about the fossils of the Burgess Shale, about 520 or whatever it is million years ago. And he wanted to ask, we're so proud of ourselves that evolution arrived at us. If evolution had a second chance, would it have arrived at us a second chance? In other words, were we somehow predestined? And he argued that if you were to wind back the tape of life, they had tapes in the days that he was writing, wind back the tape of life to the early days of the British Shale, and you just let it play a game, would it have come out to something like us? And he said no. He thought it just wasn't going to happen. So this was a thought experiment. But in fact, this experiment really has been done. Because at this point, these two are pretty much the same thing. We run the experiment once, we run the experiment a second time, and what resulted is pretty different. The outcomes are quite different. And you can imagine that were you to run this a third time, do you think either a person or a jumping spider would have come out the third time? No, not very likely. Now, I've got to admit that this isn't exactly Gould's thought experiment because, of course, these two lineages have a chance to interact. They're not on completely independent planets, right? So it's not exactly his experiment. But I think when we look at this, we realize that evolution is so full of chance that what would happen on the third or the fourth replicate of these lineages evolving and diversifying would have been very different. And in fact, it was, because there are other lineages that gave rise to other things out here. So, as an evolutionary biologist, one of our questions is, to what extent is there predictability in evolution? Is evolution all chance? With each species, a unique outcome. This sense of the uniqueness of species is deeply ingrained in a taxonomist psyche. It leads us to respect and to celebrate each one. And I want to just focus for a moment on this word celebrate. And this is a moment at which I'll go a little away from the scientific perspective. This is a plate from a taxonomic monograph that came out of my PhD thesis. Starting so early forced me to build a relationship with my subject without much guidance. No one told me how to be a scientist, right? I was 13 years old, I didn't have a professor there, whatever, but I really wanted to learn. But I wasn't thinking, this is the way that a scientist should behave. So I could approach my subject as a whole person. I could approach it as in a standard scientific way, but I could also approach it in a way that was much more about appreciation and love and how I thought about these spiders. So for me, they're not just study organisms. They're also things of beauty. And one of the ways that I expressed that in my work was that I did drawings that were combined both scientific documentation and an expression of the art that's intrinsic to the organisms themselves. So these are the oldest surviving drawings that I've got from when I was 14 years old. And then as the years went on, I got a little bit better at my technique of drawing. And so this was a few years after that. And one of the things that is clear from the way that I used to do drawings, I don't have nearly as much time as I used to, so they tend to be a little hasty these days. But they have superfluous, in a scientific sense, superfluous detail or superfluous color, even though it's true. If I was simply documenting this for a scientific purpose, I wouldn't have put these little shadows and that little highlight. I just would have shown where the hairs are. But I wanted to do this in full color because I wanted to celebrate the beauty of the spiders. There's a dual message here in what I was doing. And that's true even when it started to get a little bit more spare and technical in the black and white illustrations for my monographs. So for me, this is a celebration of found art. Every time I do a drawing, I feel like there's art there already, and I'm just somehow capturing it and displaying it. I walk into the forests not only as a scientist, but also as an artist. And I'm not alone in this. Scientists have a rational motivation to seek truth. It's how we teach, it's how we think, it's what we want to get towards. But underneath that, we have an emotional motivation to seek beauty. There's no doubt most biologists, especially ones who study biodiversity, have this attachment to the organisms. Okay, Next, we're going to start thinking it's not just uniqueness. So these are the peacock spiders of Australia. You may have seen them on YouTube. They're somewhat famous. The males, these are all males, and the abdomen, the rear end of the male, he sticks it up in the air and does courtship. And they're often full of colors. And so here are some displays of the males taken by Maddie Gerard at Berkeley. They're beautiful dances that the male does to the female. Here's the female here. Some of them are rather silly looking. And this one they gave a special name to. Sparkle muffin? I don't know where that came from. The males do such silly things to attract the female's attention. But it turns out that even though the peacock spiders are the most famous jumping spiders, there's a comparable group in North America called the paradise spiders, which are not quite so intensely colorful, but they're just as complicated. And it's the genus Habernax. And here are some courtship behaviors in Habernax. I call this the orchestra conductor species. In fact, in a talk I put this to Beethoven's violin concerto, and it actually works pretty well. So you'll notice that as he's flicking his front legs, that's up here, he's moving his palpi, the feelers, and he's going like this. Okay? Now this is one population. If you go to another population, it's a bit different. Notice how the palps are moving alternately. Left, right, left, right, left, right, left, right. So that question, do they make their motions at the same time, left and right together, or alternately left and right, is something that shows up in these paradise spiders. So here's one that's very cleanly separated, left, right, well, right, left. So these squares, by the way, are 1 millimeter. So that shows you how big they are. And this guy likewise is really cleanly. He sort of slowly waves the palp on one side and then slowly waves it on the other side. So these guys are waving the palpi alternately, but this one does it with its first legs. And this one does it in a more subtle way with the first legs. Notice how it's going left and then right on the Now this one's waving the third legs. The third pair, remember they have 4 pairs of legs. So he's shaking the knees out on one side and then the knees out on the other. Likewise alternating. So many species of paradise spider have this asymmetrical left-right motions, where something happens on the left side, then on the right side, and back and forth. And when we started finding this out, I talked to people who study the interaction between males and females and what females choose. And they were a bit surprised because it turns out that there had been these ideas that females should choose symmetry. Now, of course, summed over time, these are symmetrical, but even still, this was intriguing to us. And so we wanted to know what would happen, what would we learn if we mapped them on the phylogeny. So this is the evolutionary tree of branching lineages. What if we were to mark this up with where these asymmetrical motions are? And that's in these bars. And you can see, for instance, by the pink, that asymmetrical motions of the palp evolve 1, 2, 3, 4 times separately, on separate lineages, in different subfamilies, so to speak. So these asymmetrical motions evolve frequently. And at this point, it turns out, this presents a challenge to biology, but we haven't taken it any further yet because this is not the sort of biologist I am. I find these patterns, and then I hope somebody's going to come along and want to figure out why exactly this happens. Why do they evolve these asymmetrical motions? But I'm going to tell you a couple of possible explanations that could be looked at. One is that it's just easy for evolution to do this. These spiders already have the neuronal circuitry to walk, for instance, which is a left-right alternating motion. You want to attract the female's attention by waving something around. Why not just sort of duplicate some of that circuitry and repurpose it for this, in which case it just, as soon as it starts off, it's left, right, left, right. That's a possibility. Another possibility is that the male wants to show that he's in good shape. He wants to show that he's got high quality so that she knows that her genes will be in babies that survive well. Right? She doesn't want to waste her eggs with a bad male. And so it may be that his ability to separate these in his pal also is indicative of how well he'd run and how well he'd catch prey. The last one is a little bit different, which is that it may be that he's just confusing the female. He goes, look over here. Oh no, look over here. Oh no, look over here. And you can actually see the female sort of looking like this, and that may actually serve a useful purpose of keeping him still long enough until he can communicate other things he needs to communicate. So the purpose of this in the talk is to say there is a pattern here. Something is happening repeatedly. We don't exactly know why, but it happens again and again and again. Another pattern that's repeated has to do with color, which is that if you look at ornaments, in other words, fancy things that the male has, that are near the face, they're often colorful. So many times, colorful ornaments have evolved near the face. But if you look at the ornaments at the tips of the front legs, they're almost always black and white. And we think that that's because the female is looking at the tips of the front legs with her little eyes, which don't see color. But she's looking at his face with her big eyes, which do see color. That there's a sensory constraint in the female that's driving this pattern. So evolution isn't all about uniqueness. There is repetition, there is pattern. And it turns out that there's phylogenetic repetition in chromosomes as well. Now, this is a part of the talk where it gets the most technical, so I'm just sort of going to whiz through this a little faster than I normally would, and people can ask me questions. So it turns out that we have Y chromosomes in the males of humans, right? XY male, XX female. Spiders don't have Y chromosomes. They're just 2 X's in the male, 4 X's in the female, so that they figure out what they are by how many X's they've got. That's just the way it works. But there are a few Habernatus that have Y chromosomes. And it turns out that we can see that they evolved by fusions between the X chromosomes and the regular chromosomes, the autosomes. And if you get one fusion, you get XXY. If you get 2 fusions, you get triple XY. And as an evolutionary biologist, I wanted to know, how are these scattered across the phylogeny? And when you look at this, the white is the without Y, and the green and blue are the with Y. You see the green and blue are scattered all around. And in fact, we can reconstruct that this Y chromosome has evolved independently at least 8 times, probably more like 12, maybe as many as 15 times. And this is an extraordinary number for any group of animals. There's no case that I've been able to find in the literature this dense for such fusions. But there's something peculiar, which is that this doesn't seem to be just chance fusions. And here's the reason. What fusions do we see in general? I've focused on these that have to do with X chromosomes, but there are some fusions possibly between autosomes, although it's not clear, so I say 0 to 1 because of ambiguity, and there might be fusions involving the X's. But even if you take the most generous perspective on other fusions, it turns out that there's a really strong bias towards this type of fusion. Because if 2 chromosomes were going to fuse randomly, you pick 2 chromosomes out of a hat, you're going to get 2 normal chromosomes, not an X chromosome, because there's lots of the normal ones, but only 2 X's. So basically, the chance that this bias would have happened just by randomly is very, very low. So something is biasing towards fusions involving. So why would there be a bias involving sex chromosomes? And you could imagine something that has to do with some peculiar mechanism of sex chromosomes and what persists and so forth. But it turns out that in the literature, there is a theoretical reason that has been proposed a while back as to why these fusions might be favored, why evolution might propel such fusions. And it's sort of a weird reason. And we don't always think that our genomes could have genes that males and females among us would dispute. So imagine that you're a spider and a mutation comes in to make a red face. If you're a male, you go, oh, I'm so happy, I've got a red face, I can attract the attention of the female. If you're a male, you say, I don't want a red face. I want to just sort of hide in the bushes here, right? I don't want something to notice me. So there can be an allele of a gene, a variant of a gene that's good for males, and a different one that's good for females. And if you have a situation like that, where a mutation comes in, say, for a red face, it's potentially a problem if it's expressed in both males and females. And under that circumstance, the theory suggests that it can favor an X autosome fusion, because when that happens, you can isolate the one variant to males on the Y chromosome. The females don't have to have it. And so potentially you can have it such that the males can be happy with their nice little red face, and the females can be happy they don't have to have a red face. This is going to depend upon a lot of details, but this is a theoretical possibility as to why you would end up getting such fusions. The one thing for this is that recombination in this region must be low for the conflict to be resolved. And in fact, in general, it's been thought that for these sorts of fusion products, it makes sense to limit recombination here, because if there's high recombination, you're going to get chiasmata that are proximal. This is a little bit of geek speak here. They're going to be bunched up when things try to separate, and at least the old ideas were that that could lead to bad segregation and basically a lot of wasted sperm. But it turns out that in fact, those species that have a Y chromosome tend to, in evolutionary terms, have the chiasmata towards the tips, suggesting that they may have limited recombination in that proximal area, which means that they really are isolating that variant to males, which is sort of cool. So the other thing that this model requires, and this is something that we know very little about, that when the variant comes in, it's expressed both in males and females. And sometimes it might not be, sometimes it might luck out and only one of the sexes it shows up in. But in other cases, maybe it's in both. And we do see, in fact, some females that have traces of the courtship ornaments of their males. So these guys don't have an eyebrow. This male has an eyebrow, and you can see the same eyebrow in the female. The other thing is, and this is where the fanciness of the spiders comes in, is that every sexually dimorphic trait is an opportunity for conflict, and there are many, which is to say there are many opportunities. And so now I'm going to show you how complicated the courtship behavior can get. These are the first legs, the third legs, the face. There are many little bits and pieces that are probably under separate genetic control in these spiders. That's the morphology. Oops, just a minute. I'm going to show you this, I'm going to show it, but I'm going to actually quickly act it out because it's a lot better that way. So, this is where I wish I had a lapel mic. Okay, so the male's going to stand there like this with his palpi above his legs, and he's going to bounce his whole body up and down as he's doing this with his palpi. And he's going to stand really high. He's going to move the palp like this. He's going to move the legs like this, and he's going to sort of dive down like this and suddenly go. So that's some really weird set of things he does. And then later he's going to be sitting there doing little tweaks with his first leg, and then every so often he's shaking his third knee out on one side and then the other. And he does that for a while, and then he shakes both of his third legs out and starts this really fast thing where he's going on his right side and on his left side, and it's just sort of crazy. So here it is. Okay? Now he's shaking his knees on either side. This is clipped, highly edited. And now he's going to start flicking one side, that's his left. Now he's going to do his right. Now, she had other ideas, but you don't have to worry, males are very good at getting away, so he did not suffer in the end. So when we tried to describe this, how do you describe this? Like, how do you publish what they did? We thought, well, is there dance notation for 8 legs? Like, how would we do this? So we invented this little seismograph thing of the different body parts and how they move. So these are the first 3 stages of courtship. So we started to compile what the whole courtship is, like what's the whole song, so to speak. This is the 4th stage. This is the 5th stage of courtship. This is the 6th stage of courtship, the 7th, the 8th, and the 9th. It's stunningly complex. It's as complex as a bird of paradise, and this is in a 5mm spider. We talked about it as if it was a symphony with movements and themes and motifs and so forth. But it turns out there's actually also a song. These are the soundtracks that he's doing. And he's doing it with his abdomen, and they're little buzzing noises, and this is what they sound like. We had no idea that this was happening in people's backyards. I don't know what Stendilaurus falcerti, the one with the red face, does, but I bet when somebody puts a microphone to it, it's going to be doing something like this. So courtship involves so many traits. These are sexually dimorphic. There are many opportunities in haberdashers for sexual conflict within loci. And for instance, it turns out that it looks like most of the origins of the Y chromosomes are in the group of haberdashers that has the most complicated display. So this is our hypothesis that sexual selection drives sexual dimorphism in courtship traits. There's this intralocus conflict when new variants come in. It selects for chromosome fusions, and then either, we don't know whether the cart comes first or the horse comes first, the chiasmal localization follows or precedes. So there is repetition in evolution. There's repeated evolution of asymmetrical motions and color biases in courtship, repeated evolution of chromosome fusions consistently biased to X, correlated with these chiasmatic distalizing. So there is repetition and pattern. We can start to look at general laws. So this is an opportunity to look towards the standard narrative of science. If there is this repetition and pattern, though, how do we reconcile this with unpredictability? And really, it's not that hard. We know this in the sciences. Even though the fate of any single lineage might be unpredictable, an ensemble of lineages can show a pattern and behave predictably. You probably would have learned the gas laws at some point in good old PD equals nRT. The behavior of a single gas molecule is unpredictable. But if you put enough of them together en masse, they gain predictability. But species are not like gas molecules. This is a problem. Once you've seen one O2, you've seen them all. In fact, that's a fundamental idea. That's one reason that physics is so focused on laws. But it's also the reason, the contrast that species are not like gas molecules, is one of the reasons that we can't think quite like a physicist as a biologist. Once you've seen one species, you're just getting started. The delicate contingency of evolutionary paths makes each species unique. Species diversity has high dimensionality, is one way to state that, and each species adds new information about evolutionary processes. And just think about what each species gives us, both knowledge and to the world. Each species gives us data on what evolution can produce and has produced. It's an actor in Earth's ecosystems. It's evolved solutions to problems that we might share. So this is a drop of venom on this spider trying to look ferocious. You may know that people are bioprospecting in spider venoms for neuroactive chemicals because, of course, they've been experimenting, modifying the nervous system of insects for the last 300 million years. Maybe they found some solutions that we would care about from a medical point of view. Each species also has a beauty all its own. So now I'm going to put this together, the idea of uniqueness and pattern, to think about the music of evolution. I drew this probably in about 1985. It's the naughty bit of a male jumping spider, and I remember when I drew this, I was fascinated by how rhythmic and beautiful it looked. How it looked like an abstract sculpture almost. Just this swooping curve and this sort of thinner area and so forth. But then I put together such drawings for a whole set of species into a plate, And I stood back and I thought, it's not just beautiful singularly, species by species, but it's beautiful as diversity. There's something like a dance going on here when you look among species. And in fact, that's the way we can think about this. This is like a dance of diversity. We wouldn't see that if we only had one. So this dance of diversity is something that we study as evolutionary biologists, and one of our important questions is, to what music do they dance? Which is to say, what tempo, what melody, what harmonies, what repetition, and what surprises. And let me try to unpack that a little bit. There are these patterns that are repeated. In the chromosomes, we've got evolution of Ys and so forth. In the courtship behaviors, we have these repeated patterns showing up. And we suspect that, and in fact in the chromosomes we can to a certain extent show, they're not one-offs, they're not single characters, they're suites of characters that come in sequence potentially. And so in this case, for instance, it may be that there are sensory biases that precede, which yield the courtship traits selected for, which generates interlocus conflict, which leads to chromosome fusions, and then chiasmal localization either facilitates or follows. So there's a sense of sequence here, and each of these things, therefore, is like a note in a melody. And it may be, and this is the idea with the chromosomes and the courtship, that these melodies are played again and again each time a Y chromosome evolves. So this is a symphony through evolutionary time. We can think about the beauty of individual organisms, but if we stand back and think about diversity, we start to feel this pattern of evolution and these repetitions and these sequences, and it's really stunning. It's one of the things that drives us to see this grand sense of pattern, and because it's changed through time, it's like music. Now, I remember when I first thought in these terms about 10 years ago, I thought, oh, I'd so much love to hear this. What does it sound like? What if you were to somehow translate it to music? And then I realized, I'm a programmer. I can make this happen. So, I don't know if this will work, but so here is the sound of chiasmal localization evolving. So we're going to go from left to right, moving forward in evolutionary time, and see what we get. Okay, so this is chiasmal localization. Something weird that happened, which is that it seemed to get busier, right? And the problem is that we only have the species that survived to the present. Presumably there were lots of things that branched off and were dead ends. They went extinct. And one way to fix this busyness increasing through time would have been to have done simulations of what might have been in the past. But they didn't do that. Okay, so we'll think about that. And next, we'll do the Y chromosome responding. So here's the Y chromosome responding. So one note is a gain of a Y chromosome, one. At this point, when I was doing this, I was starting to think, I don't know if this is going to really be so amazing as I thought it was going to be. But, so now let's hear it all together. Like this from the 20th century, you know, but it really wasn't what I was hoping for. And then I realized part of the problem with this is that every lineage is using the same instrument. So you can't hear the voices separately in their melodies. And so what I should have done probably is have it so that at the root of the tree, it splits between the woodwinds and the brass or something. And then it splits between the strings, and then in the strings it splits between the bass and the smaller ones, etc., etc. And maybe we could follow them, but it didn't quite work that way. So there you go. But evolution, like music, is a crazy and sensible balance of repetition and uniqueness, pattern and chaos, the inevitable and the unexpected. And biology really needs to focus in both senses. The particulars, the rich diversity of species and their history, but also the general processes, how it all happened. And I'm going to end quickly on beauty. Why should we speak of beauty? So I'm a biologist. I normally, it's not like I talk about beauty all the time. So why am I doing it? These are species that I think are particularly beautiful. And when I find them, when I found these, I've thought, am I the first person to see these species alive? You go into some remote forest, and you might be. And I say this not to be proud of how amazing I am, but because it's such an honor to have seen such a thing. But I also think, will I be the last? So I think any of us who has any interest in biology, anyone who visits this museum and looks through the Life Through Time exhibit, probably has imagined, oh, wouldn't it be amazing to go back in time to see the living, big, non-avian dinosaurs? Wouldn't it be amazing, right? Or the trilobites or the seed ferns or whatever it is that's your favorite thing that's now extinct. And so here's Calvin, of Calvin and Hobbes, taking photographs. He changed his cardboard box that was a transmogrifier into a time machine. He went back and he took photographs of the dinosaurs, and he claims it so that he can sell the photographs and make lots of money. But we know actually it's because he just thinks it would be so cool to see these dinosaurs, right? So I think this is a common fantasy. In 500 years, we may very well be in a post-biodiversity world. And my guess is we're going to wish we had a time machine to come back to now. The Earth's ecosystems are remarkably intact at this moment. We still have 90% of what we had 10,000 years ago, probably, right? In 500 years, we may be down to 5% of it, right? And we will look back on 2019, or whatever year it is that the curve suddenly goes, and say, oh my gosh, wouldn't it be great to have a time machine to come back to now, to see these things that we just think of as everyday things, but will be like these amazing dinosaurs to those of us 500 years from now. So I have a confession. I'm actually a time traveler. I came back 500 years. I got a grant. I managed to come back. I have unfortunately, I can't go forward in time, so I have to say goodbye to all my friends and family. And my mission is the jumping spiders. There are a couple of others. I won't tell which ones they are, that are likewise biodiversity explorers that are time travelers. So my mission is basically to go out, as long as my career lasts, to find these spiders and document them, and somehow, just like you would do with a dinosaur, get as much as you could forward in time. Maybe it'll be a photograph, maybe it'll be something else. But what I tend to try to send are these. The preserved specimens in natural history museums. A natural history museum is our time machine. That's the best time machine we have to send biodiversity into the future in a way that isn't simply an ecosystem, which we may have to do. So this is what I use as a time machine to go forward. And I'm trying to make sure that we at least have these, but of course it would be much better to be able to send this into the future. Right? This is what we really want to send into the future. The problem is that when you hear about people talking about we should do this, we should do that we should feel guilty about this or whatever, we might hope that the ethics would drive our desires, but I'm too cynical to believe that works. I think it's actually the other way around. I think we retrofit our ethics to fit our desires. So to fix things, we got to actually change what people want. We got to change desire. And not, single-use plastics and all that, but actually what that they want biodiversity, that they see it as valuable. So our best hope lies in our falling in love with this beautiful earth. And that's why I talk about beauty. Not just, but the problem is, the people that know this best are the biologists, right? We go out there and we see this beauty all the time, but we need to talk about it more. We need to convince that this is also beautiful, not just among our colleagues, but to the world at large. So to be scientists, we must share the truths we uncover, but to be responsible, we must share the beauty we behold. And there's a lot of beauty in India. So despite the leeches, the Western Ghats are incredible. They're an amazing place. And I've only looked at them, I've only trekked through them a little bit, but I'm looking forward to doing more with Kieran and other Indian biologists. And my perspective on this is that we need to know more about them, and we need to know what's out there so that we can celebrate them. To preserve it for future generations, it needs to be understood. At least understood that it exists. And to understand it, increased scientific capacity in India needs to be developed. It doesn't make any sense to rely upon people from the outside. So, acknowledgements for my visit here, Krishna Kunte at NCBS for facilitating my visit, Kieran for taking me around and sharing his wisdom and helping us both learn together, and these collaborators. And I thank you for coming and listening this long.